Hi everyone, and welcome to the FSL course practical on using ICA, independent component analysis, to pre-process and analyze resting state fMRI data. So I'm going to give a very quick overview of the contents of this practical. Here on the right I have the FSL course homepage open in Firefox, and then on the left I have a terminal where we can run commands. So I'm just going to scroll down and find the link to the ICA practical. So here you can see that the practical covers much the same content that the lecture did, although where the lecture was fairly heavy on theory and light on practical details, the practical is where we actually show you how to perform these types of analyses using FSL. So we have a couple of sections on how to use ICA to perform cleaning of single subject fMRI data. And you may remember from the lecture that if we want to perform group ICA, the first thing we need to do is to transform all of our functional data from subject space into a common standard space so that we have anatomical correspondence across all of our subjects. So we have a section on how to do that. Then we have a section on how to actually run group ICA using Melodic. The next section is a bit of an aside, which just gets you to look at how the dimensionality which you specify, i.e. the number of components which you tell Melodic to decompose your data into, affects the results. When you're performing ICA at the single subject level for the purposes of denoising, the best option is to let Melodic automatically estimate the number of components, which was covered in the lecture. But when running a group ICA, it's far more common to pre-specify the number of components which you want to decompose the data into. Depending on the type of analysis you're performing, you may want to decompose the data into a low number of components, or maybe to a high number of components. And so that's what this section is all about. Then the final section is on how to use dual regression to perform a statistical analysis and to compare these whole brain resting state networks across groups. And so there's a lot of text in this practical, very similar to the other FSL course practicals, and I strongly encourage you to try and read through all of it and to really try and understand what you're reading rather than just to blindly follow the instructions, because you'll get the most out of it by really trying to understand what you're reading. So the first section starts at the beginning and it sort of walks you through what you would do if you have a raw data set, and at the very least you'll usually expect to have some resting state fMRI data, a T1 structural image and a field map image. That's hopefully the bare minimum that you'll have. Throughout most of this practical, many of the commands which we're telling you to run would actually take too long to run within the time that we have available. So what we do is we walk you through how to set up these analyses, and we give you the commands that you would need to run them. But instead of actually running them to yourselves, we will also give you the data that is produced as a result of running the commands, rather than you running them yourself. So at the very beginning, we're just going to have a quick look at some raw data. And just like with the other practicals, anything which is in a grey box is a command that you can copy and paste directly into a terminal. So this data set contains some resting state fMRI data. It contains a structural image that has already been brain extracted for you there. And it also contains a field map. And if you have some data like this, you can use the Melodic GUI to perform a single subject, basic pre-processing and single subject ICA. And the practical will walk you through how to use this interface to set up this single subject ICA. If you've done the task fMRI practicals, this should look quite familiar to you because the Melodic interface is very similar to the FEAT interface. We have a tab which allows you to select your input data and your output directory. We have a prestats tab which allows you to select standard pre-processing steps such as motion correction and spatial smoothing. And we have a registration tab which allows you to specify registration from functional space to structural space, and then from structural space onto standard space. And so the practical will just walk you through setting up this interface. But because we're going to be moving on to showing you how to run group ICA, we've already performed single subject ICA for a set of subjects. So now I'm just going to move into another directory, into one of these pre-generated data sets. So if you've used the melodic interface to pre-process and to run ICA on some data from a subject, it will possibly confusingly save the output in a directory that ends with feet. But if we go inside that directory, then we will see another directory inside it called filteredfuncdata.ica, and that's where the primary outputs of melodic reside. The main file which melodic generates is called melodic.ic.noi.gz, and this is a 4D image which contains the spatial maps that your data was decomposed into, the independent components. So I'm just going to very quickly look at that in Fossilize. And I'm using a little feature in Fossilize which will called this AD flag, which will just automatically set up the color maps for me and save a little bit of time. So there it is. And so this file is a 4D image where each 3D image contains the spatial map of one component. And so this one, well, it looks like maybe it's noise, I'm not quite sure. And you can see that every single volume within this 4D image contains a spatial map from a different component. What we can also do in Fossilize is open up the time series view. And so this contains the time series that corresponds to this component. And so now that I have the time series up, I'm starting to think that this particular component is probably neuronal in origin because it's localized to grey matter and it contains a fairly smoothly varying time course. In contrast, a component like this is probably going to be driven by noise because most of the contribution to this component is actually on the boundary of the brain, and the time course is full of high frequency fluctuations. So that's just an example of what melodic will actually output when it's run at the single subject level. And in the next section of the practical, we're going to get you to look at some data sets like this and to manually classify some of those components. So again, you're going to use Fossilize, and we're going to get you to look at two different data sets. One of them is a multi-band acquisition and the other is a more traditional single-band EPI acquisition. And you're going to manually label a small set of components and decide for yourself whether each of those components represents real neuronal signal or whether they represent noise. Once you've done that, once you've labeled every component from your single subject ICA, you may recall from the lecture that the next step is to take the time courses of those noise components and to regress them out of your original fMRI data. And what that will do is the residuals that are left after you've regressed those noise components out will be your denoised or cleaned fMRI data ready for analysis. So in FSL, we can use this command called FSL regfilt to do that for us. And the practical will show you how to do that. And then it will get you to look at the difference between the noisy fMRI data that you started with and then the denoised fMRI data after you've regressed out all of those noise components. Then there's a short section just discussing the automatic classification tools that were mentioned in the lecture. So the next section contains a bit of technical information about once we've performed our pre-processing at the single subject level, we will then want to take all of our cleaned fMRI data and pass it to a group ICA. And before we need to do that, or before we can do that, we need to transform all of that data into standard space. That's what this section is about. There's also an optional section at the end which goes into a bit more detail on how you would go about doing this. And then this section explains how you would actually run how you would use Melodic to run a group ICA. We don't actually run it because it would take too long, but we have basically, if you end up wanting to perform this on your own data, you can come back to this practical web page and use it as a reference. So you can see, given your data, if your data looks like this, then we give you the command to run it on your own. And then, as I mentioned earlier, there's a little section on which compares a group ICA which has been run with a low dimensionality with one which is run with a high dimensionality. And you'll see that resting state networks which were decomposed into a single component in the low dimensional ICA will have actually been split up into multiple components in the high dimensional ICA. And then the last section talks about, once you've done your pre-processing, once you've run group ICA to identify your resting state networks, you can then use dual regression, which will relate the group ICA components back to each individual subject and will then allow you to compare the contribution of each subject to that group level network and to perform statistical analysis. And we do that in FSL using dual regression. So this section of the practical just steps you through that and gets you to have a look at some results that we've already generated for you. And that's it. I recommend that you ask the tutors questions whenever you're unsure about something. We're here to help, and chat with the other course attendees either by voice or by online chat. And above all else, just have fun. Thanks.